Fouling communities are communities of organisms that live under docks and ship hulls. They contain a variety of invertebrates, algae, and microbes. These communities are strongly impacted by the environmental conditions and human activities in that area. The goals of this experiment were to track changes in fouling communities over time, specifically how introducing new species to an area changes the number and types of organisms in the community. This allows researchers to see how different species interact and gives them a chance to detect invasive species before they become problematic. To collect data, researchers suspended plastic tiles from the edges of docks and piers so they can hang upside down in the water. Each tile hung for 3 months. During that time, organisms attached and started growing. Scientists would then collect the tiles for photographs in the lab. To participate in this project, I had to go to the Zooniverse website. I clicked on Invader ID and then clicked Classify. This is a picture of a PVC panel that they used. I have to zoom in because they want me to classify what's in that circle. I go over to the lists and see what I think it is. I go through the pictures, then read the description. Seems like it looks about right. So then I'm going to scroll down, click Identify, and Next. The reason I decided to choose this project was because as a coastal environmental science major, you learn all about the negative impacts invasive species have. For example, we have the brown and knoll here in Florida. We also have the lionfish. On this project, I was able to find out about Didynum vexillum. This invasive species is from Japan, and it rapidly blankets hard surfaces, making it difficult for the members of the fouling communities who survive. I chose this project because I wanted to do my part as a scientist to ensure that we have a healthy marine ecosystem. Since I didn't grow up near the ocean, I'd never heard of a fouling community. This project taught me not only what these are, but also how important they are to our knowledge of our marine environment. They act as quick indicators if there is an introduction of an invasive species and provide nurseries to crabs and larval fish. Participation in this project helped me better understand how interconnected our ecosystem is. These organisms in the fouling community act as food and nurseries for several various marine species. If these communities go ecologically extinct due to anthropogenic activities, it'll have devastating consequences for our marine habitat. By participating in this project, I'm able to assist scientists in monitoring these communities to protect coastal areas and to help respond to these invasions more effectively, which would protect numerous species from extinction.